Welcome to IQ Checker YouTube channel, home of science quizzes and IQ tests.
This is paper 2, multiple choice questions for the extended candidates. So we'll be taking this as part of preparation for this year's October-November IGCSE Biology. So from here we'll move straight to question 1. Question 1 says, some processes are carried out by living organisms are listed. 1. Egestion, 2. Excretion, 3. Reproduction, 4. Respiration. Which processes are carried out by all living organisms? So from all these processes, you can observe that egestion, not all living organisms carry out this, but excretion is carried out by all living organisms, reproduction by all living organisms, and respiration by all living organisms. So according to this, we will see that the only option that is correct here is option C. Option C is our correct answer. So we'll now move to question 2. Question 2 says, the scientific name for humans is Homo sapiens. What does Homo refer to? So, in scientific naming of living organisms, this Homo is the genus, while sapiens is the species name. Right, so automatically, Homo refer to not mammal, but the genus, not kingdom, and not species. So B is automatically our correct answer. So we'll now move straight to question 3. Question 3 says, some structures found in cells are listed. Number 1, we have the cell membrane, cell wall, mitochondria, and the nucleus. Which structures are found in prokaryotic cells? Prokaryotic cells, number 1, they lack the nucleus. Alright, and the mitochondria are not found, not in all prokaryotic cells. So we have the cell membrane and we have the cell wall. So automatically that makes option A, 1 and 2 as the correct option. So B is wrong, C is wrong, and D is wrong. So we'll now move straight to question 4. Question 4 says, in a mesophyll cell, where are chloroplasts found? Chloroplasts are always found in the cytoplasm of the cell. It is not between the cell wall and the cell membrane. No, that's the intermembrane space. It's not in the nucleus. There we have the genetic material and the chromosomes, not in the vacuole, but in the cytoplasm. That makes B our correct answer. So now question 5. Question 5. Question 5 says, the diagram shows a flatworm. The flatworm has been magnified by 100 times. You can see the length, that's the image size. What is the actual size of the flatworm? And according to magnification, magnification is equal to image size over the object size. Alright? And if you look at the options are in micrometer. So these are value here. You convert 50 millimeter to micrometer. So you are going to multiply this by 1000. So that will give you 50,000. Alright? So our magnification here is 100 is equal to the image size, which is 50,000, over the actual size, which is the object size, which we don't know. So making our object size the subject of the formula, or the actual size. So actual size is equal to 50,000 divided by 100. So with this, we'll now have this cancel this. We'll now be left with 500 micrometer. Alright, so that makes option D our correct answer. 500 micrometer. That is the actual size of this flatworm. So question 6. Question 6 now says, the diagram shows how a cell appears under the microscope at the start of an experiment, and after it has been placed in a dilute solution of salt for 5 minutes. So the process, this is the starting point, having this result at the end of the experiment. So which statement explains what happens? So if you look at this, you can see that this cell placed in the dilute salt solution has increased in size. So this increment is as a result of movement of water molecules into this cell. Definitely water molecule has moved into the cell. And the movement of these water molecules is as a result of osmosis. So it is not dissolved salts entering the cell by diffusion. It is not dissolved salt leaving the cell by diffusion, but it is rather water enters the cell by osmosis. That makes option C correct, and water leaves the cell, the option D is wrong. So that makes option C the correct answer. So with this, now we move to question 7. Question 7 says, the concentration of nitrate ions is higher inside the root hair cell than in the soil. What would be required for the absorption of nitrate ions in the root hair cell? We have mitochondria, oxygen, membrane proteins, and cell wall. So in this case, nitrate ions are higher inside the root hair cell than in the soil, and the plants still need to absorb this from the soil. So it is definitely going against a concentration gradient. And for this to happen, you require energy. So because of that, mitochondria will be involved, oxygen will be involved, and transport proteins, which are the membrane proteins as well, will be involved. But the cell wall is not necessarily required. So with this, we have 1, 2, and 3. So our option A here is wrong. Option B is our correct answer. Option C is wrong. Option D is wrong. So 1, 2, and 3, option B is our perfect answer here. So we now move straight to question 8. Question 8 says, what is the color change shown by Benedict solution when heated with a reducing sugar? Benedict text with reducing sugar. When reducing sugar is present, it always changes from blue to red. Benedict solution is a blue solution. So it's not blue to purple, but it is blue to red. Not brown to blue-black, and blue-black is even test for starch. Red to yellow. So our correct answer here is option B. So option B, blue to red, is the test for reducing sugar. Now question 9 says, the diagram shows the digestive system. Which part absorbs the most water? So the part that absorbs the most water, so we just need to label these parts first as quickly as possible. The part labeled A is the stomach. We have the stomach here, and we have option B, the large intestine. Okay, the large intestine. Option D here is the small intestine. Right, the small intestine. And option C is the anus. And in the digestive system, most absorption takes place in the small intestine, and the same thing goes with the absorption of water. The absorption of water takes place the most in the small intestine. So that will now take us to question 10. Question 10 says, which statement is correct? The active site of an enzyme has the same shape as the substrate. Of course, that is wrong. The active site does not have the same shape as the substrate, but rather it must be complementary. So option B says the active site is not affected by changes in temperature. It can be affected. When it goes beyond the optimum temperature, it can be denatured. So option C says the shape of the active site of an enzyme is complementary, and that was what I just said earlier. They should be complementary. Then the substrate, that's option D now, the substrate and product combine at the active sites. The substrate and product, no, but rather the product is released from the active site. So that makes option C our correct answer in this case. So we'll now move to question 11. Question 11 says, the diagram shows how the rate of photosynthesis of the plant changes with light intensity at 3 different carbon dioxide concentrations. In each case, the temperature is 15 degrees Celsius. What is the limiting factor for the rate of photosynthesis at point X on the graph? What is the limiting factor? Alright. In this case now, the light intensity is the same. And obviously, the only thing that will affect the rate of photosynthesis here should be the concentration of carbon dioxide. They are exposed to the same light intensity. Alright, so it cannot affect it. Surface area, they are using the same plant, and not temperature. In each case, they are exposed to the same temperature of 15 degrees Celsius. So the only factor here that can affect it is the carbon dioxide concentration. That makes option A the correct answer. So we'll now move to question 12. Question 12 says, the diagram shows a test tube at the start and at the end of an enzyme experiment. At the start and at the end of an enzyme experiment. So at the end of the experiment, the mixture in the test tube contains glycerol. So glycerol is produced as a product. Okay, glycerol is a product here produced. Which row correctly identifies the enzyme and the substrate it acts on? So before we even go to the options, you just have to, first of all, glycerol is produced. Which substrate will produce glycerol? Definitely the substrate will have to be a lipid, otherwise known as fats or oil. Okay, definitely it has to be a lipid. Then the enzyme that breaks down lipids are the lipase. So with this now at the back of your mind, you should be able to just move straight to pick the correct answer. So we have option A that says amylase as the enzyme and fat as the substrate. Then we have option B that says amylase and glucose as a substrate. Option C says lipase and fat as the substrate. So remember, it was said that the product is glycerol. So definitely option A is wrong because of the enzyme. Involved. Option B is wrong because of the product involved. But option C is correct. Why? Because the enzyme is lipase and the substrate is fat. So all the options are correct. But option D is wrong because the substrate is not glucose. So this makes option C our correct answer. So now move straight to question 13. Question 13 says, the diagram shows the teeth in the lower jaw of a human. Which tooth is a molar? So definitely we just have to label these parts. Part B is called the incisors. Only the incisors. We have the canine, right? Then we have the premolar. And option D. Automatically option B is our correct answer. Then question 14 says, what is meant by chemical digestion? So, if you really understand what chemical digestion is, you should know that in chemical digestion, large insoluble molecules, take note of that, insoluble molecules are broken down into small soluble molecules. That is chemical digestion. It is not large soluble, no. It is not small insoluble, no. And it is not small soluble. Alright? So that makes option A our correct answer for question 14. So at this point, we are going to stop this video here. If you have not subscribed to our channel, please and please do well to like and subscribe to this YouTube channel for more updates. So we'll continue in the next video. Thank you.